Tell us a little bit about your position at the university and your areas of interest.
So I'm working at the Institut de la Vision in Paris. It's an INSERM and CNRS and University Institute. And we are working on the identification of mutation in known genes and as well as in novel genes by using targeted and whole exome next generation sequencing approaches. And our focus is on congenital stationary night blindness, a non-progressive retinal disorder, but as well as on more progressive retinal disorders like rod cone dystrophies or cone rod dystrophies. And furthermore, we are also genotyping patient with Stargardt disease for gene therapeutical trials. Thereafter, we are interested, if we identify, for example, a novel gene defect, to see with all the data which is created today with next generation sequencing, you can find more than 50,000 variants. So it is very important to identify the disease-causing mutation which underlies the phenotype. So then you need to validate which is the real gene to be implicated in the disease. So what we are doing is we are using bioinformatic screenings. So there are transgenic mouse animal models available where you can indeed see if your gene of interest is expressed, for example, in the photoreceptors. Then it is a good candidate to be implicated in photoreceptor degeneration, like we can find in rod cone dystrophies or in other disorders affecting the retina. If you have, for example, a gene candidate for congenital stationary night blindness, then you are more interested in genes which are expressed in the inner nuclear layer, especially in the bipolar cells. And then there are other models via bioinformatic tools we are using, like transcriptomic databases available to us from our collaborator, Thierry Levillard, which are based on RD1 mice. RD1 mice are mice where the photoreceptor degenerate, especially the rods, during age. And so if your gene of interest degenerate as well, or the expression gets lower with age, then of course you can also estimate that you have a photoreceptor specific gene, and then you can see, that's indeed a very good candidate. Otherwise, around, if you are interested in a CSNB, congenital stationary night blindness gene, then the expression needs to be increased because we don't have any more photoreceptors, but bipolar cells are still present, so you have the impression the RNA is increasing. But however, we are also a research lab, so we are doing also proteomics. So we have access, luckily, to human and mouse retina. And today there are many different antibodies available for all genes and proteins up to the market. So we order these antibodies and test if the proteins are indeed localized in the tissue we are interested in. And this makes them a good candidate, but nowadays we are also interested to go further to see, to understand the pathogenic mechanisms of the newly identified genes. So we have done this especially for, more recently, for a protein implicated in rod cone dystrophy, cilia protein. Even if you have all these strong hints that this is a good candidate gene, then often a second family is missing, having the same mutations. These days we will only identify rare disease-causing genes. Most of the disease-causing genes, like usherine, which affects more than 15% of the RP patients, have been already identified. So we are really looking for the last genes. But still, about 40 to 50% of the cases miss genes in the known mutation in the known genes. So we estimate that a lot of more genes needs to be identified. Or that these variants, these disease-causing variants, may be located in intronic regions or on RNA level. something which needs to be developed in the future.
Sounds like you're running an extremely busy lab. You're covering a number of very critical and important bases. What are some of the most interesting areas that you've discovered, or the material that you're either publishing or you're currently working on?
For me, the most interesting work is still considered the work on congenital stationary night blindness, because it is an amazing disease, because it's non-progressive. So you can learn a lot about the signal transmission from photoreceptors to bipolar cells. As you may know, photoreceptor transduction cascade, there is a lot known already, but and of course it's always good to investigate the proteins which have been associated with the disease by performing mouse modeling and so on. But for congenital stationary night blindness, this is really a perfect model to study the signaling from the photoreceptors to the bipolar cells, and the signaling is yet not as known as the phototransduction cascade. So in this respect, as soon as we have identified to date, I think 8 different genes underlying this disorder, it helped really to dissect the signaling pathway. So you have the metabotropic glutamate receptor, and now every year new components can be added to this. We have done this work, gene identification, mainly at the beginning with classical linkage analysis. Later on, it was really an exciting time when we have performed candidate gene approaches, where in the literature, for example, a horse model was described showing also night blindness and decreased expression of a candidate gene. And finally, we screened, due to this horse which has had night blindness, we screened this gene as well for mutation in patients, and indeed were able with others to identify TRPM1 mutations leading to night blindness. And so candidate gene approaches are worthwhile, but today with the demand and the availability of NGS, it helped us as well to add 2 novel molecules to this cascade. So a lot of molecules are now identified, and we would like to go further. And by doing in vitro analysis, but also in vivo modeling, to really see the function of these molecules. So my PhD student, for example, she's working, Marion Noyer, she's working on a molecule LRID3, and this is important obviously for this channel TRPM1 to bring it at the membrane on that it can function, and then that the people can see during dim light conditions. That is very interesting, and we even started now a project for this disease on therapy. We are doing an ARV replacement therapy approach for mGlu6, which is one of the major gene defects underlying this disorder, and think it's a perfect model. So CSNB is not only a perfect model to understand the signal transmission in these cells, but also to find therapies, because this is a non-degenerative disease. So in principle, you can treat patients at any time, because the photoreceptors, the bipolar cells are still there, but it's a more functional problem. And of course, because the retina is an accessible organ. So.
Tell us about the translational implications. You've done a remarkable amount of work in the laboratory, identifying variants, identifying candidates, working on the proteomics. What are perhaps the earliest translational implications of your work?
The first work we started is now on mGlu6, one molecule implicated in congenital stationary night blindness, and we are interested to treat patients with this form of CSNB. Another one.
The European Union recently commenced its first gene therapy trial in humans. Are you familiar with it? Are you involved with it?
I'm not yet involved in that, but I think it depends really on the model and the disease you are using.
What is the position in the European Union? I know we're still waiting for the FDA to approve the first clinical trial in any application. How receptive is the European Union to accelerated clinical trials for specific areas?
Bad question for me. I guess I'm not really.
You've just moderated a very important session. What were the highlights for you?
So I was really surprised how many people are working on iPS cells today, and also there were really promising results how iPS cells can really give now the shape to be a real retina, especially in forming the RPE cells and also the photoreceptor cells. And also the procedure, I learned myself a lot about this technique. How then thereafter you need, of course, if you're taking a fiber or skin and make fibroblast and make then iPS cells, that you, of course, if you want to treat yourself, you need to make also gene editing on these iPS cells. So I was really surprised how many different steps are implicated in this work, which then will hopefully help to treat patients. And then you have another step, which is do the photoreceptors integrate then to the retina, the existing retina. So I think it is really exciting, and it was represented in a really exciting way, but I think it will still take some time until it goes to clinical trials, as it has been shown for the AAV approaches. And I think it's really interesting. It's very good to do this work, and especially for dominant diseases where you have dominant negative effects. However, AAV strategies for small genes, especially, I think it's a little bit more advanced. But I think it's very good to speak about these new techniques. But it's a lot of work.
Finally, tell us about your next steps in your laboratory. What's on the horizon for you for the next year or so?
So we would like also to continue with our therapeutic approach, so especially, as I mentioned, congenital stationary night blindness to treat, because it is really an interesting model, and also to start with iPS cells. However, here we have still limiting factors, because the bipolar cells, you can make nicely now RPE cells and also photoreceptor cells. For bipolar cells, our colleagues in our institute have shown, Olivier Gouraud, that it is possible, but it's limited. So I like to work on iPS cells to better understand the pathogenic mechanism as well for CSNB, but also for RP. We have started already on this work to study the pathogenic mechanism and also for the treatment. But of course, I think the iPS cells of the retina needs to be still improved to resemble more the patient or a human or a mouse.
Professor Zeitz, thank you so much for a very illuminating and insightful interview. And we wish you all the very best of luck in your research going forward. Thank you.
Thank you very much.
Yeah.